So in contrast with mitosis, where you start off with your pairs of homologous chromosomes and you end up with your pairs of homologous chromosomes, i.e. you start with diploid cells and you end up with a perfect copy of those diploid cells. The point of meiosis is to generate gametes. Now, let's start with a bit of terminology. So let's just get our bearings. So for what we're talking about here is we're talking about homologous pairs of chromosomes. Homologous chromosomes. They are two sections of DNA, one that you got from your father, one that you got from the mother, and they have the same genes on them, but possibly different alleles of those genes. Okay, so we can say if we had a section of DNA here on a homologous chromosome or homologous pair of chromosomes, that section of DNA would code for the same protein, similar proteins. Okay, so the proteins that do the same job, I should say. All right, so they might be both sections of DNA that code for a protein that gives the person the characteristic of eye color. But those two different chromosomes may have different versions of that gene. So from the father's side, this gene might code for a protein that results in someone having green eyes. But from the mother's side, this chromosome might, and this allele might code for a gene product that would result in the person having brown eyes. And depending on which characteristic is recessive or dominant or how those genes are interacting with each other, that will result in a person having a particular characteristic. Okay, so that's important that we understand what we are dealing with here. So homologous chromosomes, they have the same genes, but possibly could have different alleles. Okay, they might even have the same allele. That's also possible. So when we refer to people being or an organism being a heterozygous or a homozygous, we're referring to the position, the genes, the alleles at a specific position. Okay, all right. So homologous chromosomes have the same genes, but possibly different alleles, even possibly the same alleles. Okay, all right. So homologous chromosomes, we would refer to these two as genes or loci. All right. So genes will be in the same, genes for the same thing, controlling the same characteristics, should be at the same loci on homologous pairs of chromosomes. And we can refer to these also. So those two together are genes, okay? But if this is A, this could be little a, and therefore different alleles. Okay, so we refer to this as different alleles. Okay, and depending on the combination of alleles, individuals will be homozygous or heterozygous if they are different. Okay, homozygous if they're the same, heterozygous if they're different, and depending on if they're dominant and recessive, that will also come into it. Okay, so I think, yeah, that gives us our bearings. So just remember, you've got two of these homologous chromosomes, and one is essentially from father's side, the other essentially from mother's side. Okay, right? And and depending on the combination of genes, certain characteristics, certain phenotypes will develop. But the focus of this is not that, but it's this idea that how meiosis can work to generate more variation. Okay, I'm not going to go through the individual steps of meiosis. I think that you can do yourself. But what I will do is look at the process and how this process is, how it occurs, and how it generates variation, how it increases variation. Okay. And remember, generating variation is very important for the evolution of species, for natural selection to occur, for species to be able to adapt to changing environmental conditions. If you've got lots of variation, lots of combinations of alleles, different combinations of alleles generating lots of different types of phenotypes, if you've got lots of variation within a population, that means if the environment changes for whatever reason, if the environment changes, there'll be lots of variety. Some of those variations will be adapted to the changing conditions, whereas others might not. But if we don't have variations, the conditions, the environment might change, and none of our population might have any adaptations to that environmental change. So to have variety in a population is very important for natural selection to be able to occur if environmental conditions change. Okay. All right. Or even if the conditions stay the same, for populations to continue to become better adapted to those conditions. You actually got variations. The best ones will always survive because they'll be selected. They'll be the best. They'll have the best combinations of alleles and genes and mutations. They'll have the best phenotypes to be able to adapt better to that environment than the other members of the population, and therefore they can survive better and pass on their genes to the offspring and the next generation, via their offspring to the next generation. Whereas if you don't have variety, you might not be able to, or the population might not be able to continue to adapt better to its environment, or if the environment changes, they might not be able to adapt to that. Okay, so we're going to be looking at this process and how it generates variation. Okay, so let's begin then. Let's look at a particular locus and the alleles that are there. We could have, for example, eye colors there. So we can say that the locus for eye color is here. Eye color is there. And on a different homologous pair of chromosomes, there is the locus for hair color. Hair color. So we're just going to take a very simple example and use this. So shall we say E and E and H and H. And let's just set a few rules down. So for the capital E, let's say that E is dominant and it gives you brown hair, brown eyes, and little e is recessive and it gives you blue eyes. And with hair color, the capital H will be, say, black hair, and little h, recessive, blonde hair. Okay, so this individual is heterozygous for eye color and heterozygous for hair color, but the phenotype that they have is they are an individual with brown eyes and black hair. Okay, now what we're looking at in this process of meiosis is how this individual in their body, in their reproductive organs, they are producing gametes. Okay, so from a normal diploid cell, we're starting here, right? So this is an important stage, diploid. Now in a human being, Homo sapiens, our diploid number of chromosomes is 46, i.e. 23 homologous pairs. All right, in different organisms, there's different numbers of those pairs. But anyway, our gametes have simply 23 chromosomes and not 23 pairs. Okay, so the chromosomes get halved in the gametes. Okay, and we call them haploid as opposed to diploid being a normal cell. Haploid gametes. All right. So say this individual is a man. These are the possibilities of his sperm in a very simple one scenario. Okay. So let's just look at how that happens. So in the first instance, what happens is that each of the homologous pair of chromosomes will replicate, duplicate, and form this. And this is called a bivalent. Okay, so this is called a bivalent, and that is an important term. Bivalent, right? So they each replicate, and once replicated, they are joined. You know, the two identical copies of this chromosome, joined at the centromere. So this is now called a chromosome, centromere here, but that chromosome is two copies of the original chromosome. The other chromosome also replicates, and those two copies are joined at the centrosome there, but these homologous chromosomes are connected to each other physically, and that whole thing there is called a bivalent. So that's one bivalent, and that's another bivalent. Okay? So we've got two copies of each homologous chromosome here, two copies of that homologous chromosome there, and so on. There will be 23 pairs in a human being. Okay, but we're just looking at two of them. Okay, so that occurs there, and then each of the bivalent is then pulled to opposite sides of the dividing cell, and we get that situation. I hope we can see that split apart from that, those split apart from each other, and we get this situation. So let's just see at this stage where the alleles have ended up. Okay, so the two big E's have ended up over there, and the two little E's have ended up there. Hope you can see that. Whereas for our hair color, the alleles have ended up like so. So the two dominant alleles have ended up in that cell, and the two recessive alleles, have ended up in that cell. Meiosis is two divisions. So main point of the rest of the stages is that we divide these chromosomes further. Okay, so remember, this is actually two copies of each allele. Now we don't, in our gametes, we only pass on one allele of each. So these chromosomes each will undergo further separation, and let's just follow how this will happen. So this cell will undergo its own division. Each of the different chromosomes will line up. They will all be separated because remember, each different chromosome contains the very important genes required to make that full organism. So we can't have one chromosome, one entire chromosome going in one cell and this chromosome going in another cell, because both the cells then will be missing one of the chromosomes which very likely does contain important genes to make the organism. So each pair is split apart so that each gamete contains one copy of all the genes necessary to make an organism. Right, so let's follow these genes then. We have our E here, E there, and H here, and H here. And here we have our recessives. There. So I hope you can see that what we will end up with is gametes, 50% of gametes that will be, whoops, like this. And the other 50% that will be like this. Okay, and that's a very simple case. So, yeah, so we've ended up with, let's just review. We started off with someone who was that, and that's diploid. They've got 2 copies of each gene, 2 alleles of each gene. Via meiosis, we've generated sperm cells which now only have 1 copy of each gene. Okay? And this is important because each of these sperm will fuse, well, not all each of them, but we know one of them will then fuse with an egg. And that egg will also have 1 copy, but depending on the genotype of that person, you know, they'll have different alleles. When they come together, the resulting zygote will again have 2 copies of each gene, each allele. Sorry, each gene, yeah. Okay, and that person, thankfully, will be diploid and will then, you know, have their own set of phenotypes determined by the combination of genes in their cells. Okay, and then they will go on to make their own gametes, and the circle of life continues. Okay, so that's a very simple case. But what we now need to look at is how this process, what is it about this process that leads to variety? Because this is not a lot of variation. Where does all that variation come from? So I'm going to get rid of all this again, and now we're going to look straight at how we generate different types of gametes. How can we increase the number of combinations that we've produced here? Okay, so the first way is the first thing is called independent assortment of chromosomes. Okay, independent assortment of chromosomes. We've looked at one scenario. It could have happened like this, but it could also have occurred in a different way, which is if this bivalent had arranged itself like that. Now let's just look at what effect that would have had. So this one would have been here, and what we're saying is that this one could have been there. Still homologous chromosomes, still the bivalent, but the bivalent could have assorted differently because it's independent of the other chromosomes. It's not physically linked to the other chromosomes, and so it could have assorted in this way. It could have organized itself, arranged itself in this way. And if that was the case, how would it have affected the gametes that would have been produced? So if this would have happened, and the likelihood is that, you know, it does happen, so while sex cells are being generated, while meiosis is happening, in some cells they assort in one way, in other cells they'll be assorting in other ways. And in those cells, let's see what kind of gametes we would have generated. So in this case, we would have ended up with our H here and our capital H on this side. That means that at this stage, should I just change everything and well, actually go a bit on? So at this stage, we would have ended up with the other chromosome, this chromosome on this side, this chromosome on this side, okay, which means that here we would have ended up with a different combination of chromosomes, alleles in each cell, because this cell would have had this one here, chromosomes getting increasingly dodgy, and this one here, which means in this cell we would have ended up with our H's, recessive alleles, and in this one we would have ended up with our dominant alleles, and that means that later on we would have ended up with a different situation here, because, should I carry on in the same way? Because, I'm sure you can see where this is going, because we would have ended up with these guys here, this being the H, capital H is the dominant alleles, and this one here, this being the recessive alleles there. And that means that we would have generated a whole other set of gametes. Shall I just change them? I'll just change them. Which means that that would have ended up with there. Okay. So we ended up with something like this, with the recessives here and dominant alleles in this. So these gametes would be passing on a different combination of genes, okay, which would have produced a whole different individual with different characteristics than if it was one of the other sperm that had fertilized the egg, okay? And so we've seen how independent assortment of chromosomes leads to a different set of alleles being passed on. We've increased the possible allele combinations, and therefore we've increased the possible number of the possible combinations of phenotypes that there could be. Okay, so yeah, independent assortment of chromosomes. That's one. Now, the other thing that could happen is crossing over, or chiasmata forming. Okay, so crossing over via the formation of chiasmata. Okay, all right. So in order to talk about crossing over and what effect that can have on the combinations of alleles that we could possibly be generating in our gametes, and leading to obviously the increasing genetic variation, key term there, genetic variation. What we're going to do is we're just going to add or look at another allele on the same chromosome as our first one, right? And say that's D, and I didn't really think this through. I don't know what phenotype it's going to code for, or yeah. But let's just say that it's there, and not really too bothered with what it codes for. But it is a phenotype in the organism, all right? Remember, this chromosome is going to have lots of genes. Anyway, now, if all of this happened without crossing over, the allele combinations that we would have got are like so. So D would have, little d would always be with the big E. Okay, they are linked. D, little d, the recessive D is always linked to the dominant E allele. Okay. And the big D is always linked to the little e. So wherever there's little e, because it's on the same chromosome, this will always be big D at the bottom right there, okay, at that locus. But what we're saying is that if crossing over occurs, that combination could be altered and new combinations could occur, i.e. new combinations of genes, alleles, sorry, could exist. And how does that happen? So when these bivalents form, sister chromatids right here could, sorry, non-sister chromatids, not sister chromatids, non-sister chromatids could exchange DNA. Okay, remember we said that there was a physical connection between this chromosome and that chromosome. There's a physical connection there. And that physical connection can sometimes lead to the swapping over of whole entire sections of DNA. And so what would occur here, and they have to be non-sister, because remember, if the sister chromatids exchange DNA, there would be, because that DNA is identical, because it was produced from replication, the crossing over between sister chromatids would generate no new allele combinations because they're identical. So we always talk about the crossing over between the non-sister chromatids. So I'm just going to put that there. Crossing over between non-sister chromatids. So I'm just going to draw what that would look like. What that would mean is DNA, corresponding DNA, remember they got the same genes, only different versions of the gene. So corresponding DNA from this chromosome would move to that chromosome, and from this chromosome to the other chromosome. So what would that look like? It would look like that. But that doesn't really mean much until we now say that on this chromosome right there, instead of now it being linked big E to little d, it's now linked with that one, because the DNA is swapped over. Now the gene, the specific gene that codes for the dominant allele, has now moved over to this chromosome, and from that chromosome to the other one, only in this one. The other two chromatids are the same still as before. So here we have the big D now linked here with the big E. And on this sister chromatid, we still have the E, but it's now linked with little d, the recessive allele. What does that mean? That means that in two of these gametes, these chromosomes are going to be altered so that this, so this one is going to be with not big D, but little d, and this one will be with big D, okay? Because the crossing over has occurred here. So this one ends up with that little red bit there, and this one has ended up with that little blue bit there, okay? So this doesn't always happen. This doesn't always happen, and it's likely that it can happen in different places. So we have now generated a whole, independent assortment led to an increase in our possible allele combinations, but crossing over mixes that up more, because now we've got new allele combinations. Before, without crossing over, E was always a big E, the dominant E was always linked with the recessive D, and that's the genes that will be passed on, all right? So if that D was, I don't know, skin color, what a bad example. If that D was big feet, then that means that, someone with brown, the allele for the dominant allele for brown eyes would always have inherited the allele for big feet. Okay? But now, because of crossing over, that's not the case, because in some cases gametes will have crossing over had occurred in that process of meiosis, generating different combinations. So now it's not necessary that someone with the allele for brown eyes has the allele for big feet. They might have the allele for now small feet on those chromosomes. Okay, so independent assortment of chromosomes, crossing over generates further genetic variation in the way that we've just seen. Another thing is that there could be also, and I don't need to go over this again, but there could be independent assortment of the sister chromatids as well. Independent assortment and segregation. And segregation of sister chromatids. So just like we said that, depending on how the bivalents organize themselves before they are pulled to opposite poles of the dividing cell, in the same way, it does make a difference where the sister chromatids end up, especially when they have crossing over has occurred, because it makes a difference whether the crossing over, whether the sister chromatids with crossing over, where the crossing over has occurred, which other genes do they end up with? Okay, so that also makes a difference. Yeah. So other things that could happen are mutations, and mutations can occur before all this happens. When the DNA is replicating to form these bivalents, there could be other rearrangements of the DNA that could occur. We're not talking about single base mutations that could occur also, but we are talking about large arrangements of DNA where there could be whole sections of DNA that are kind of amplified, or there are whole sections of DNA that are inverted, or whole sections of DNA that are just deleted. All these things are possible, and they all add to the variation of gametes that can be produced. Finally, fertilization. Fertilization. So remember that fertilization increases variation, because you can have any one of these gametes fuse with any one of a number of different possible eggs. Okay? And when that occurs, remember that these haploid gametes are fusing with other haploid gametes to form a diploid zygote. And that diploid zygote will have more combinations of alleles than haploid cells can have. Okay? So even the process of fertilization itself, the simple act of fusing haploid cells increases the phenotypes that could be produced. Yeah? So fertilization itself increases genetic variation. Okay? And that is it. I hope I have made sense of a few things, but essentially realize that meiosis is the process of halving the kind of genetic information in a way, all right? And if you really just want to simplify the process down, if you've got homologous chromosomes, just draw a line here and you've essentially done meiosis. Okay, that's meiosis right there. Some cells, some gametes will end up with this, other gametes will end up with that, all right? But you don't know whether the pink one was on this side or the blue one was on that side. So if the pink one was on that side and the blue one was on the other side, then our meiosis has just, we've increased the number of variations. Okay, and yeah, sometimes genetic information can swap from one chromosome to the other, and that's essentially crossing over, right? It essentially unlinks genes that were previously linked, and that increases the variation. Just as these bivalents can independently assort, so can the sister chromatids at this stage. So at that stage, independent assortment meant that chromosomes can end up in different sites. But in the same way, these sister chromatids can end up in different cells depending on how they arrange themselves on the equator during that stage. Okay, and finally, mutations. Another big part, mutations can occur to increase the variation as well. All right, guys, if there's any questions, let me know. Good luck.